Thanks for having me. So I'm going to walk you through one of our research projects. That's development of precise and equitable diagnostic kits for early detection of COPD. Yeah, let's start with some numbers. And I'm actually from Kashmir, and we're a very small population of 3 million people, and we're located in the northern Himalayas of India. The air quality index is very good in Kashmir. But when I joined SKUAST, I was surprised to know that in our population as well, we have almost 300,000 to 500,000 patients, known patients with COPD, and that was kind of surprising to me. And when I look at the national index, in India, we have almost 7 to 10% of the populations with COPD, and moving to the global population, we have 500 million population with COPD. But what actually happens in COPD is it's a progressive and irreversible lung disease, which is characterized by the continuous lung distress. Actually, what happens in COPD is a patient is unable to breathe normally. What happens here in us actually, when we breathe, air goes down from our windpipe to our lungs, and in lungs it moves from different airways, the network of airways, and at the end of each airway, there's a small air sacs, okay, which holds air. And when you exhale, those air sacs deflates, and that's a normal procedure. But in COPD patients, you can see on the left side of the slide, those airways actually get blocked. They get thinned, and there's a lot of inflammation in those airways, in those airways, because of which the air could not flow properly. And these airways actually generate a lot of mucus, which further limits the flow of air. Okay, and these patients, COPD patients, they are unable to breathe normally. So this is just a basic introduction about COPD and how. What are the major causes of COPD? Cigarette smoking, obviously that's the #1 reason of COPD. Apart from that, secondary smoke from biomass burning and other things from industries, that's a secondary reason for smoke, your air pollution. And another is genetic predisposition. Few people, like, they are genetically determined to have COPD, but that's very uncommon and that's very rare actually. So now, as we know what is COPD and what are the causes of COPD, now let's move to what are the diagnostic tests for these COPD, current diagnostic tests. First is spirometry. In this spirometry test, doctor actually gives you a kind of a tube, and the patient has to blow through that tube. It's a lung functioning test actually, how good your lungs function. But to my experience, this spirometry test it's confounded by so many factors, and it does not give you a right picture of COPD, whether a person has COPD or not, or at what stage a person is. The next is chest X-ray, digital X-ray, or something like that. But again, there are so many confounding variables in chest X-rays as well. To my experience so far, what I have seen is CTs gives you good results, but the problem there is it's high cost. People don't afford that, and patients get exposed to radiations. And in COPD, you have to do repeatedly tests to see whether even lifestyle interventions makes a positive impact or not, or whether a medication impacts your COPD or not. You have to do it repeatedly, and that's not good sign. So globally, researchers are looking into differentially expressed genes, microRNAs, non-coding RNAs, some histone modifications, and DNA methylations as well, to see whether it's possible to find certain markers for COPD. So my research focused on DNA methylation. DNA methylation is actually a small epigenetic modifications on cytosine basis when it's followed by guanine in a DNA sequence. What basically this DNA methylation does is it tells different enzymes how to read DNA sequence. Okay, that way your genes get expressed or something like that. And now our study is, I'm just briefly discussing this thing. We have recruited 2,300 patients from four different ethnic groups. We have Indian population, around 1,200 samples, European population, Pacific, we have patients from Pacific, New Zealand and Australia, and Asian population as well, like 300 to 400 samples from Han Chinese and others. So what we did is we collected their phenotypic information, high-resolution CT scans, and everything we collected from those patients. And from there, we went from Illumina 850K array sequencing. No, not microarray, sorry. And now this is the results. We got 9 genes that are hypomethylated between the cases and controls. Remember these genes, these 9 genes at the top figure, these are in different populations, and we combine the samples, all 9 genes were persistently hit. You can see at the top hit, there's AHRR gene. AHRR is known to be associated with smoking. So when we saw this figure, we realized like, oh, maybe we're not seeing COPD versus non-COPD. We're looking at smokers versus non-smokers. We did a lot of linear regression, some rank regressions, and we corrected for smoking, and we identified 3 genes that are strongly associated with COPD. So when we found these 3 genes, you can see in these 3 genes, all the genes are hypomethylated. That means in control samples, the methylation level was close to, say, 0.8 or 0.9, while as in COPD patients, the methylation levels goes down. It's hypomethylation in patients. So from these 3 genes, we decided, like, is it possible to develop any diagnostic, methylation-based diagnostic kit for COPD or not? So for that, we did ONT sequencing. We multiplexed our samples using adaptive sequencing. We sequenced only a portion of the genome. Using one of our collaborator, Dr. Hina Khan, she's a molecular biologist. She standardized sample multiplexing and adaptive sampling technique. And we have found the results that we got from Illumina that were very consistent with actually ONT. We got exactly good results. And the risk group, case group, and control groups that were differentiated properly between all these 3 sites. Moving to this slide is very interesting actually. You can see the top 4 patients. The methylation level is 70%. This is from ONT. The methylation level was 70%. They were healthy controls, no COPD. But here, below this point, these are active smokers from 13 to 24. Those are active smokers, and they are known to have a COPD. But when people stop smoking, so you can see their level goes up. Their methylation level goes up. We have dig deep into this, and we have seen the people who have never smoked, or they have a methylation level of somewhere close to 70 for all those 3 genes. While as the smokers, they have a methylation level of close to 25 or 40. But here's the reward. When a person quits smoking, or when the lifestyle interventions happens, then the methylation level goes up. And when we have followed these patients, the number of allergens actually that happens every year, the number of cases reduce drastically. Kashmir is a temperate region. We have a long and very harsh winters, and all these patients, they complain for actually allergies when the winter arrives. But when because of lifestyle interventions and when they quit smoking, the number of cases reduced dramatically, and their symptoms got relieved as well. So yeah, that's it. That's it from my side. Thank you very much.